Okay? And it would have an intrinsic GTPase activity causing GTP hydrolysis and inactivation. These processes, the guanyl nucleotide exchange and the GTP hydrolysis, would be mediated by a guanyl nucleotide exchange factor and a GTPase activating protein. That's the normal G protein cycle that we've seen. And is it the normal cycle that we see with the small G proteins? This is again another illustration of that cycle. We have guanyl nucleotide exchange factors mediating guanyl nucleotide exchange, and GTPase activating proteins enhancing the GTPase activity. So it's a reversible activation, deactivation process. Okay. And RAS is a small G protein. And yes, this is part of the normal RAS activation cycle. This is the normal RAS activation cycle. It is activated and deactivated in much the same way as we've seen for other G proteins. So the first human oncogene turned out to be a new class of G protein, a small G protein. Now, I'd like to just talk about a couple of very interesting mutations that were found in this G protein. One is an activating mutation. Okay. And this is similar to what we've seen with the toxins' effects on G proteins. An activating toxin, like cholera toxin, would affect this cycle, and a mutation, an activating mutation, would similarly affect this cycle. And that active mutation would be a mutation which blocks the intrinsic GTPase activity. Okay, so if you block the GTPase activity, you remain in the active configuration, GTP bound, and you're constitutively active. So one mutation, an activating mutation, would be a mutation that blocks the GTPase activity. An inactivating mutation would be one in which the nucleotide exchange, the ability of the protein to bind GTP, is blocked by mutation. So let me ask you a clicker question, and that is, which of these two types of mutations, which of these two mutations do you think was contained in the first human oncogene? So the first human oncogene was a mutant form of RAS, and I've just shown you an inhibitory and an activating mutation. And the question is, A, is it an inhibitory mutation? Or B, is it an activating mutation that has caused RAS to become an oncogene? Does everyone understand the question? A is inhibitory, B is activating. Okay. Would anyone like to defend one choice or the other and explain why a particular mutation would cause it to become oncogenic? Yes.
I had it be the activating mutation because it stops the G protein from being activated, and it will continue to grow, and it will continue to use the GTP
Okay, good. So what he said is he believes it's the activating mutation because now you have a constitutively active RAS, and if it's involved in growth control pathway, by analogy to SRC, the active form would cause uncontrolled growth. Anyone else? Yeah.
I chose the spot activating because once you start the GDP process, so it will always be active, and then as the effector, well, it also depends on the function of the effector actually. If the effector
Yes, excellent.
the growth, then that would make it proliferate.
Okay, so what you're saying, what she's saying is, give your name. What she's saying is that it really depends on what the effector is, okay, and how that effector plays a role in normal growth control. Okay, so if that effector is a positive effector which mediates division, then uncontrolled activation of that effector would stimulate division uncontrollably. Anyone else? Anyone who picked the inhibitory? Change your mind? Yeah. Okay, very good. Very nice. So what she's saying again is it depends on what the role of the effector is. Okay? If you have, if you inhibit that ability of the effector to be functional and that effector is stimulating growth, okay, then you're going to get more uncontrolled growth. The key here, though, is understanding that it acts in a dominant way. Okay? So while you might envision either an activating or an inhibiting mutation to cause uncontrolled growth, only the activating mutation would allow you to act in a dominant way. So remember, if I introduce the mutant form of RAS, Weinberg showed, if I introduce the mutant form of RAS into normal cells containing the normal cellular form of RAS, it acts in a dominant way to cause uncontrolled growth. If that mutation blocked RAS function, then introduction of that into normal cells would be very difficult to block in a dominant way to overcome the normal growth control by the wild-type protein. Okay? Now, it turns out that an inhibitory mutant, this type of inhibitory mutant of RAS, could in fact act in a dominant way under special circumstances. Can anyone think of that? Of what circumstance? What we have is a mutation that blocks the ability of this protein to bind GTP, and I introduce it into cells. How could it block the endogenous normal cellular function of RAS? How could it be dominant? That's a tough one. Okay. Yeah. Sorry? No, we're not adding any more toxins. All we're doing is introducing this mutant into cells, and I'm asking, how could it be acting in a dominant inhibitory way? Yeah.
Is it possible that RAS could be acting as a repressor? So if it's inactivated, then you won't have checkpoints, and the cell will just keep on growing since this is not active?
Well, the thing is, the normal cellular gene is there and active. Okay, so we're introducing a dead form of RAS, an inactive form of RAS. How could it act in a dominant way to inhibit normal cellular RAS? Okay, I'm going to tell you. Yeah, one last try.